Okay, so it turns out that we've really covered a lot of the organelles, so we just want to finish up with this group we call the cytoskeleton. So it's the cell support structure. The cytoskeleton is made up of, yes, proteins, because we said proteins do all the functions. And there's three basic groups we talk about. We talk about these filaments, microfilaments. We talk about these intermediate filaments, and we talk about tubules. So these are cells, right, that have been stained with different fluorescent dyes to look at each of these types. Okay, and they form these amazing structures, right? But you'll see that the cytoskeleton is all over the place. And it's really providing the shape, structure, and it's also allowing some of the movement and assisting with cell division when we talk about it. So this group of, again, organelles, that's a loose term for them, but they are really assisting in a number of vital things for the cell, but most of it, again, is support, shape, structure, and movement. All right? And again, three basic groups of this. So centrioles, right, which you might have heard of in relationship to cell division and mitosis that we're going to talk about here in a little bit. Centrioles are formed in this organelle called the centrosome, and centrioles direct the production, the assembly, more or less, really, of cilia and flagella. Right, so cilia seen here, flagella or flagellum here, and when we see cilia and flagella, we want to think movement, because these are microtubules. They're pretty large structures, all things said and done. And they either move the cell itself like it does for the sperm, or they move something that's on the surface of the cell. And this is just a close-up or a highly magnified image of this. So they do differ in their location, length, abundance. So cilia are shorter, flagellum longer. Often there's a lot of cilia and generally only one flagellum, right? And then we move into the other requirement we had, right, for cells. Now that we've covered all those organelles in the cytoplasm, we need to talk about the command center, the nucleus. And then this will take us into cell cycle. So the nucleus shown here in a cartoon is right up against this endoplasmic reticulum and rough at that, which is very important because there's a coordination between the code for the proteins and, of course, the manufacture of the proteins. So it's got this is the nucleolus it's a structure, if you will, inside the organelle. Here's our membrane that encloses that, and of course there's pores to let things in and out of the cytoplasm to nucleus and nucleus to cytoplasm. This is how we're going to pass RNA out of the nucleus and hook that RNA, messenger RNA, up to the ribosomes. So here, again, some terms. Nuclear envelope or membrane is surrounding it and does have pores, as I say here, to allow small things in and out. Chromatin, chromosomes, this is the term for the organization of all that DNA, those nucleic acids inside the nucleus. And the nucleolus is an area where there's a high density of ribosomal RNA and ribosomes formed, the ribosomal subunits, I should say, that will eventually be exported and put back together out in the cytoplasm for protein synthesis. All right, we're not going to do a Kahoot here. We do those in the beginning, so I just haven't taken it out yet. All right, so how is DNA organized? Well, when we talk about the entirety, we call it the genome, and that's about, like I said, it's 3 billion nucleotide pairs, or 6 billion, right, nucleotides. And in all of that chemical information are sequences, usually 10 to 15,000 nucleotides, that make up things we call genes. And you become a gene because your chemical information can be used to make a protein. So we say simply that a gene is a segment of DNA that codes for a specific polypeptide. Many peptides, of course, are 2 or more amino acids. All right, the genetic code is talking about how we take that nucleic acid sequence, DNA, and use it to make a protein sequence, amino acids. All right, it turns out you have roughly 20,500 genes that make you a human being. And all that genetic information, again, is contained in that nucleus. And that nucleus, remember, came from your mom and dad. All right? So the genome, as I said before, is all your DNA. You have about, 6 billion nucleotides or 3 billion nucleotide pairs. All right, so here we're looking at this. This was, again, nucleic acids was one of the big 4 macromolecules we talked about in Chapter 2. So here's what it comes down to. This is unraveled, right? So what happens in the cell is to get 6 billion, right, nucleotides in there, you really have to do some winding, right? It's kind of like winding up, you can take 75 yards of a floss or something and put it in a tiny, inch space if you wind it, or thread, or you name it, right? Yarn. So this is the same concept, but it's very, very sophisticated, right? So ultimately, your nucleic acids are made of these building blocks called nucleotides, and so adenine, thymine, guanine, cytosine, right? They have a specific order, and it's that order that determines what amino acids are going to be made from the gene. Okay, here in your book it says 22,000 genes distributed on 46 chromosomes, but other sources are a little bit lower, say about 20,500, maybe 21,000. I'm not going to worry about the numbers. The numbers are not what's important in understanding this. It's important to understand what's happening here, okay? And as I've already said, these came from 23 chromosomes from your mom and 23 from your dad. So these chromosomes represent a whole lot of genetic information, right? So I do remind you that we have these 5 carbon pentose sugars, ribose or deoxyribose, attached to phosphates, and then that nitrogenous base, adenine, guanine, cytosine, thymine, right? There's only 4, and that's it, so you got to make everything, all these different genes out of just 4 singularities, if you will, right? The bases pair specifically, so we do understand that, so DNA is double-stranded, and you'll notice that a T always with an A C always with a G, right? C, G, right? A, T. All right, so it turns out these purines, chemical class, adenines and guanines, purines, 2 rings, always have to bind to a pyrimidine. And so we refer to that as a complementary base pairing. And then these double strands wrap around these structures called histones, proteins, and then they wrap and wrap and wrap. And they form a structure called chromatin, all right? If the chromatin wraps up even tighter, which it does during cell reproduction, mitosis, we get a chromosome, all right? So in order to produce a new cell, right, because we keep coming back to this idea that you got a whole lot of different cells now, but all of them came from that one. So how do we get here? How did we get to where we are now? So there was a lot of DNA replication, okay? So it occurs, it's a pretty straightforward process, honestly. It's got to be inside the cell's nucleus, because DNA never leaves the nucleus. It occurs when you're growing, because growth is adding cell numbers or cell mass. It occurs in some cells throughout your entire life, and it'll continue even when you stop growing. When you damage a cell, you'll have to repair cells. You have to repair and replace them, so you have to have replication. To be a cell, remember, you have to have this DNA, right? It occurs by basically unwinding the 2, right, chemical strands and building 2 new strands. So I say here, hydrogen bonds break, because that's what's really holding this together. They show you as dotted lines here. Again, thymine to adenine, guanine to cytosine, and it's between the bases, right? So hydrogen bonds break, DNA unwinds. Right, so when it unwinds the unpaired bases, these pairs are pulled apart. An enzyme called DNA polymerase will basically attach to each unwound and separate strand and start building. So each strand will get its own polymerase and start building new strands, forming new bonds, and using the original strands as templates. Right? So this is such an efficient process that in about 6 hours you can copy all those 3 billion nucleotide pairs. I say it occurs in stages. Stage 1, right, occurs with helicase, the enzyme, right, there it is, separates the 2 strands, and it's stabilized by a series of other enzymes. Stage 2 is an elongation, so initiation start. Elongation is just adding new nucleotides, right? based on what is already there because of that complementary base pairing. So elongation occurs because of DNA polymerase, right? Polymer is a long chain of this. And it's semi-conservative again because you are using an original strand to build your new one. And then, of course, when you get to the end, you terminate. There are lots and lots of safeguards, lots of enzymes and proofreading enzymes and repair enzymes that are trying to check to make sure that as you're building new strands that you're putting the right nucleotide in the right place. When you have completed this task, a cell can look forward to dividing because it has now 2 copies of the code. All right? So protein synthesis, all right, I say here that whatever the cell is doing, whatever its job is, it probably involves, right, a protein. So it needs to make, and I said really on average, cells make about 2,000 different proteins. So how do they do this? All right, well, this very simple cartoon is trying to show you this, and it's a little bit cut off here, but you've got the book to look at here for that figure. So the sequence of DNA, right, here's our DNA, double strand, turns out it separates just like it did for DNA replication, and one of the strands is used to build a new strand. And so in this case, a template, this is the strand used, the other strand just doesn't participate. And here's my complement, right? Where there's a T, I build an A. Where there's an A, not a T, but a U, because RNA has a uracil instead of a thymine. Where there's a C, a G, a G, a C, and so forth. And so you build this. Now, also remember that you only have to build as far as the gene goes, and genes on average about 10 to 15,000 nucleotides, right? So this occurs a lot faster, right? Well, we'll take it a step further, and again, the process of going from DNA to RNA, and this is specifically called messenger RNA, this is known as transcription, right? Because when you transcribe something, you copy it, right? Now, every 3 nucleotides are a code. They're a cue to the cell that represents a specific amino acid. And each codon, right, and there's 64 different ones possible, can only code, right, for one specific amino acid. So AUG is methionine, even though you can't see this, and only methionine. UCU is serine and only serine, right? And so every 3 nucleotides are known as a codon for that reason, because they're codes, right? Now remember, as I've already said, but we want to reinforce, DNA, right, is contained and maintained in the nucleus, not going to leave. Proteins, right, in this process of translation, are made by that, right, rough ER, and specifically the ribosomes. And remember, why do you need proteins? Well, they give us all of our functions. So RNA is a way to overcome this difference. And DNA stays in the nucleus, proteins are built in the cytoplasm. How do I get from DNA to protein? And again, I've mentioned RNA, and we've talked about it in Chapter 2, but we just need to kind of really reinforce that. So DNA cannot leave the nucleus, but we need that information, chemical information carried in there, to assemble the right amino acids in the right order to build the right protein. Protein synthesis done in the cytoplasm. So the solution to this problem we present here is messenger RNA. It's made in the nucleus, and it's a single strand, right? So here's the transcript being made, just like you kind of saw that on the previous slide here. All right? RNA nucleotides are complementary, as I talked about. Where there's an A, it's a U. Where there's a T in DNA, there's an A. Where there's a G, there's a C. With that exception, again, that RNA molecules do not contain thymine, so we use uracil instead. The copying of DNA in this process that goes on in the nucleus and the production of messenger RNA is known as transcription. All right? So it looks a bit like this. This cartoon is pretty good at this. Similar to DNA replication, but uses only one strand and different enzymes, no thymine. There's initiation, elongation, and termination. This can be done over and over and over, and you can make multiple copies of the gene.